Let us define first the bacteriophage. Bacteriophage are obligate intracellular viruses that specifically infect bacteria. They were discovered independently by two researchers, Frederick William Tort at the University of London in 1915 and Felix D'Herelle, who confirmed the finding and coined the term bacteriophage in 1917 and have been much studied since. The bacteriophage structure includes capsid head, nucleic acid, collar, sheath, base plate, spikes, and tail fiber. In order to reproduce, phage must first enter the host cell. They bind to specific receptors on the bacterial cell surface with their tail fibers or adsorption and create a hole, a process which along with attachment is coordinated by the base plate. Here is an illustration showing the step-by-step of lytic cycle and the lysogenic cycle. But for us to understand more, let us talk about first the lysis or known as the lytic cycle. Lysis or lytic cycle is commonly referred as the reproductive cycle of the bacteriophage. A virus undergoes lytic and lysogenic cycle to reproduce. It is relatively common wherein a virus infects a host cell, uses metabolism to multiply and destroy the cell completely. This lytic cycle is one of the two cycles of viral reproduction, referring to bacterial viruses or bacteriophages, the other being the lysogenic cycle. And the lytic cycle results in the destruction of the infected cell and its membrane. The lytic cycle, which is also commonly referred to as the reproductive cycle of the bacteriophage, has 5 stages cycle. These are attachment, degradation of the host genome, synthesis, assembly, and lysis or the new virus. The first step in lytic cycle is attachment. A virus requires a host cell for multiplication. In the first step, a virus attaches itself onto the host cell. The virus may attach itself on the receptor of the cell, or simply any other parts on its surface. In some cases, a small parts of the viruses attaches to the host instead of the virus itself. Next is penetration. Once attached the virus, they gain access to the cell body by breaking through the cell's plasma membrane. In some cases, the cell wall tail fibers of the virus and enzymes released by its help in breaking through the cell. The virus then releases its genetic material into the host cell. The cell is now said to be infected. Then we have seizure of cell mechanism or synthesis. The infected DNA or RNA then hijacks the host cell's genetic material and mangles with its gene expression. If DNA is injected, the virus DNA transcribes itself into mRNA molecules. These molecules can control the host ribosomes. Thus, the virus gains control of the host cell's mechanisms. If RNA is injected, an enzyme called reverse transcriptase transcribes the virus RNA into DNA. The virus takes over the cell machinery to produce multiple viral components. Simply put, the virus infects the host to produce more virus parts. Also, virus puts the host nucleotides to work and replicates its own genetic material. Last, we have virus multiplication and cell destruction. Now that the genetic material as well as viral parts are replicated, the host metabolism assembles new viruses from scratch. The capsid are assembled, followed by the genetic material in the head, and finally the tail fiber is attached to the end portion. Eventually, the host cell is filled with viruses. Once the assembly is completed, the virus releases an enzyme that disintegrates the cell wall from within, thus destructing the host cells. The viruses are individual and free now. Next is lysogeny or the lysogenic cycle. Referred as temperate or non-virulent infection, does not kill the host cell, instead using it as a refuge where it exists in a dormant state. It is a rare method of viral reproduction and depends largely upon the lytic cycle. In contrast, The lysogenic cycle does not result in immediate lysing of the host cell. Those pages able to undergo lysogeny are known as temperate pages. First stage of lysogenic cycle is penetration. Once the virus genetic material is inserted, it attaches itself to that of the host. Some viruses may not immediately start multiplying or replicating after infecting the cell. In case it does not start replicating, the virus is said to be dormant. The virus genetic material is known as prophage while it is in the dormant stage. Next is replication of genetic material. Thereafter, the cell will fall back into its regular metabolic activities and eventually prepare for cell division. The genetic material or nucleic acid replicates and the nucleus divided into two parts, each part having the same genetic information. Then, the last stage is cell division. The cell organelles will be replicated in some cells, followed by division of the cell body, resulting into two daughter cells, each having various genetic information incorporated into their genetic material. The cells have alternate normally until the virus is triggered. These daughter cells carrying the viral genetic material are known as lysogenic cycle. I have here a table chart showing the difference between a lytic cycle and lysogenic cycle. So before I end my report, here is a short video about lytic and lysogenic cycle, which will be posted after this presentation. I hope you have learned in this discussion. That's all. Thank you and God bless.